So the another factor also vitamins and the organic supplement, the yeast extract, malt extract, protein hydrolysis, coconut water, temperatures, illumination, subculture of the tissue, every static culture, cell suspension culture. These are the important terminologies that people are using the in vitro things. When you want to more discussion on this point, you can discuss to me. So next thing is the culture conditions are very important. So explants are very important. What are the plants are using? It's called as explants. Growth curves is very important. So the growth curve should be the sigmoid one. So because the growth curve determine the secondary metabolites. When the lag phase is scanned, no growth in the cell is adjusted the new media after the subculture. So whereas the stationary phase, the primary and cell proliferation come to halt the nutrients in the medium are exhausted. Dry weight may be declined. The cell utilizes the store the reserve material. Exponential phase, primary metabolism increase tissue proliferation rapidly with the consumption of the medium and nutrition. So this is a growth cycle. So the growth period is 1 hour, doubling the time. This various stages. So we are all discussed. So the elicitor, we should know about the elicitors. When they talk about the elicitors, this is a stress factors, abiotic stress, light temperature, nutrient, growth regulator, precursor. Whereas the biotic stress factors, biotransformation, aerobic culture, genetic transformation, these are things of biotic stress. So when the gene expression, the elicitors, when we involve into that media, automatically is defense response and gene expression will be happen. So these are the steps involved in these things. So few things I have to discuss here. There is the selected list of the plant species involved in the hair root culture. We know that one hair root culture is another technology for the production of the secondary metabolites. That means natural product. Few of the natural products is already approved by the FDA, Food Drug Administration, because it's no side effect is there. So the recommend for that people administration. So next is stress factor. This is a stress factors. There is a abiotic stress. There is a aerobic culture. The precursor, elicitors, nutrients, and temperature and growth regulators are very important for that production of the secondary metabolites in the in vitro states. So we involve the various biosynthetic pathways. So we have to modify the biosynthetic pathways. Based on the pathway, we have to get that more amount of that compound, respective compound. So this another thing is methods of secondary metabolite production through the manipulation of the nutrient improvement, sugar level, nitrate level, phosphate level, growth regulators, and precursor feeding. So optimized the culture environment, temperature, illumination, medium pH. So elicitation, fungal, bacteria, and yeast. The organ culture as a source of the micropropagation and organogenesis. The hair root culture for pharmaceutical, biotransformation, or the production of the secondary. So all the factors are various methodology the people are trying based on the lab facilities. If they have the very good lab facilities, the people are doing the biotransformation studies. So based on the studies, the people are improve their compound through the various techniques. So our studies are we have done that our PhD work and we have now doing that many of the project work in this aspect. So our one of the successful project is in vitro production of gymnamic acid from the Gymnema silvestri. This Gymnema silvestri, we know that one is Tamil is called as a Sergurjan. It is a very good traditional and very good antidiabetic medicinal plant. So we have to isolate the gymnamic acid from the Gymnema silvestri. Using the gymnamic acid from the Gymnema silvestri, we have select the material for the leaf material, but it is also available the other material, root material it is having. But when compared to the rooting material, it is having that more amount of secondary metabolite, leaf material having more amount of secondary metabolites. So it is we have taken the large quantity of gymnamic acid. We harvest the plant. We collect the plant and using of the plant material. It's every fresh material to the in vitro culture. So we have produced the callus, a different stage of the callus. So we are seeing that slide. We have different type of callus, the same leaf material but different plant growth regulators. We have getting the green compact colors, green loose colors, white free colors, white brown colors, such type of colors. Wherever it is a gymnamic acid is chlorophyll biosynthetic pathway. So when it is the gymnamic acid is colors are the green color, these are having that more amount of gymnamic acid content based on the biomass. These are the things are very important one. So follow that we have get some help from that quite a pharmaceutical university Japan, because normally we are not purchase the compound from that sigma or some other thing. First of all, we are trying to contact the different professors who are working the natural part. We are getting the standard compound from them, because they are isolating the very pure compound, no side effect, as well as they are already NMR data, IRUV, all the proper documentations are there. So we are trying to get that compound from that working professors, working labs. So in addition for that, our work, the gymnamic acid compound is a pentacyclic determinants. It is having a gymnamic acid around 18 type of gymnamic acid there. Of that, 1 and 2 and 4, 1 and 4 having that more effect than the other gymnamic acid. So this is the Professor Yoshikawa and that Arihara. These are the group members. They are quite a pharmaceutical university in Japan. The professors are giving to me the standard compound of gymnamic acid for our entire research work. They are the people are first isolating the purified the compound from that world. These are the people. When we are continuously approaching them, finally they are contribute whenever we need the standard compound, they are contribute to us. So we want to know that what is the role of the gymnamic acid. Nowadays, gymnamic acid is available in the sigma. The glycosidic gymnamic acid may block the absorption of sugar from the intestine. The glycoside of gymnamic acid may block the sweet taste of sugar. It has also shown the repair or regeneration of the insulin producing beta cells in the pancreas and balance insulin level. So we want to, this is an advantage of the gymnamic acid. So now we are seeing that there is we want to know in our mind it is inhibit the sweet or not. We want to know. So that is why the traditional people, if they are normally they are take 2 leaves and take eat the leaf after the few half an hour, 1 hour. After 1 hour, 2 hours, you cannot feel any sweet taste. If you are taken sugar material or laddu material or any other sweet material, you feel like that stuff. You feel like that soil. So what is this main goal is the gymnamic acid go into that attack into that sweet tongues. Automatically, the tongues inhibit that the gymnamic acid contents inhibit the sweet buds. Automatically, you feel that like soil. So it is a very basic work. We are determined that one. We are also tried our callus as well as our leaf material. We have successful result. So this is a biosynthetic pathways. We are screening the gymnamic acid production. There is involved in the cytosol and plastic. So there is a various enzymes are involved. Various interprecursor we are using malonic acid, those type of things. This is a biosynthetic pathway of pentacyclic determinants. So we are using the different type of enzymes that are improving that gymnamic acid production. So we have successful some result. This is a continuation of the pathways. So if you are going to synthesize the high quality of that secondary metabolites, the people are nowadays they are using the squalene synthase and the fluorosyl phosphate synthase and alpha amyrate synthase and beta amyrate synthase. These are the products also available in the sigma. So when you want to add to the particular type of the compound or particular type of enzymes into the media, automatically you can get the more amount of gymnamic acid. Example, if you are getting that one culture tube, if you are getting that only 1 microgram, 1 nanogram of the compound, 1 nanogram of the compound, using this precursor, maybe you can get the 10 microgram of the compound. The possibilities is there. So because based on the literature, based on the research survey, the people are suggesting that when you are adding the precursor, so automatically you can get the more amount of the secondary metabolites. So this is we determined. So the gymnamic acid product, the gymnamic acid product now available in the market. for the human purpose. So various gymnamic acid content added to that some particular product with some additional composition for that one. Gymnema capsules, diabetes support formula, BioSafe, and Gymnema cap, Gymnema tea, Dioda, Biodia. So many type of the products are available. I have given very limited list only. When you are go through the internet, you can get the more amount of the gymnamic acid product is available. The people are using for the diabetes. Who are having the diabetes? You are having the diabetes. If they regularly take the one tablet or one spoon like that, so automatically it maintains the. What is the role of this one? So already I told you it maintains the blood glucose level as well as it regenerate the pancreatic beta cells. So automatically it has to produce that. It is prevent that. It is very helpful for that human being. So gymnamic acid is one of the important for that curing of the diabetes and helpful. So we have that some publications also is there using of callus biomass. We have the very good publication in the Gymnema silverstate gymnamic acid. We have published more than 10 papers are we published. Of that, 8 papers are reputed journals, including the phytomedicines, agro-food industry, high-tech. So many journals we have published. So our callus by in vitro production of gymnamic acid from the Gymnema silverstate through the callus culture and biotic abiotic stress condition. This is a callus growth curve. This is a callus biomass. We have published the paper. This is a callus growth curve. I already discussed with you. We have the log phase, exponential phase, and stationary phase, decline phase. Based on this phase, we can get the 20, 15, 25, 35, and 45, and 55 are days. So based on the days, we have get the biomass. Based on the biomass, we can determine that stationary phase having that more amount of secondary metabolites when compared to that exponential and decline phase. Decline phase, automatically the cells are going to die. So it is a very important. So we have to add to that specific concentration of the things. We have to determine those things. So the followed that the callus culture using we have used the specific concentration after optimization. We are determined the using of color lights, green, red, blue, and white light, all the things, as well as temperature 35, 30, 35, 20, 25, all the degree temperature we are using. Then carbon source 2%, 3%, 4%, 5, 6%. After 5% having the better result. When we are studying the photo period, 4 hours light, 8 hours light, 12 hours light, 16 hours light, and that 20 hours light, 24 hours complete dark condition. The 12 hours light having the more potential to synthesize the gymnamic acid. And nitrogen sources 1 micromolar, millimolar ammonium nitrate, 2 millimolar ammonium nitrate, 3 millimolar ammonium nitrate, 4 milligram micromolar ammonium nitrate. We have used of that 3 millimolar micro having that more amount of gymnamic acid synthesis. So from the studies, we are determined that the green color callus and the maximum dry weight biomass at 45 days having the potential for the production of the gymnamic acid. This paper are already published in the reputed journals, in the phytomedicine, such type of journals. So we have concentrated after the purification, isolated, and the compound determination of gymnamic acid. We are doing that as a pure compound as well as our callus indexed compound. How much the pharmacology studies have been successful. We are determined those things. So we have to publish those things of biomedicine and phytomedicine, such type of journals. Automatically, it is we have the biomedicine. We have published the blood glucose level and how much it is involved in this pathways, such type of things. Then in vitro callus and leaf extract, gymnamic acid, stimulate the beta cell regeneration and antidiabetic activities in blister rat. Such type of thing we have discussed in this paper. So in addition that the gymnamic acid, when we are working on the obesity aspect, it is excretion of the fecal cholesterol. So when the administered into the gymnamic acid, the animals will be the animals fecal material was analyzed. The analyzed materials normally if we are having the obesity people, automatically unwanted cholesterol will be excreted by the fecal material. That's why the Japanese and the Koreans, they are everyday life, they are using the Gymnema tea. What is the use of Gymnema tea? Is if they are having any obesity, the extra cholesterol if their body they have, so automatically it will be extracted by that fecal material. So this is a gymnamic acid advantage. So it involved that pancreas regeneration process. So so this is our animal model. We have checked with the animal model. We are given the oral. We are isolating the pancreas. We are isolating the liver. We are subcutaneously inject to the animals. Such type of things we have done successfully. This is pancreatic beta cell regeneration. We have pancreatic beta cell regeneration in the different cells, different rat models. We have sectioning and determination of things. This paper is published in the Phytomedicine. So summary and conclusion, so we know that one natural products are based on the presentation. Natural product chemistry is a very important one. It is helpful to derive the new compound as well as unknown compound, as well as already is approved compound. We have synthesis large quantity through this methodology. Second thing is the biotic and abiotic stress factors are important because when we are normally plants having the very less quantity. So if the less quantity is secondary metabolite, people are having more people are having the cancer, diabetes, more amount of people have the various diseases. So in this condition, the very limited secondary metabolite is not sufficient. So biotic abiotic stress factor is very helpful to improve the large quantity of the secondary metabolites. So from the observation of gyptic acid production, the growth curve is sigmoid 1. So log phase was observed in 15 days. Exponential phase is 25 days and 35 days. Decline phase determined observed in 55 days. So known compound are improve the large scale through the in vitro condition recommend to the pharmaceutical company. So the in vitro compound through the isolation, then pharmacological studies, then it is approved going to the approved to the pharmaceutical industry. In addition, I want to tell few things. So based on the previous slide, you have to take, don't avoid that, don't take unwanted foods. You have to take that food who are making the home, who are making the people are who prepare the home, the food, the healthy food, vegetables, fruits, and daily life. If you are taken, you can prevent that many of the diseases, including COVID, including heart attack, including that, including diabetes. All things will be automatically prevented. Don't go to the junk food. Junk food is always creating the many of the issues. We know that one. We have understand last 2 years who are having the junk food. They are affecting more than the traditional food. So you have to improve your immune system. Thank you, one and all.
Thank you, Doctor. Thank you for wonderful presentations, and you give me a clear idea for the phytomedicines, how to prepare a phytomedicine, how to develop a phytomedicine, how to design a research for phytomedicines. That it should be very useful for us. Thank you, Doctor, for this wonderful presentation. And I have one suggestion, Doctor. Can you share your international experience? Actually, where you go and what experience, international experience?
So you mean that the international experience, yes, sir?
Yeah, Doctor.
Okay. So first, after completing my PhD, I went to that South Korea. I worked there in 2 years. It's a Marine Bioprocess Research Center, Korean National University, Busan. So there I was working 2 years with the Korea Science Engineering Foundation Fellowship. The lab is completely regarding working on the seaweed. So the parallelly the labs having nearly 60 students. They are research scholars. 60 of the 10 are postdoctoral. I am acting as one of the postdocs in this lab. So we have the separate building, the same building, adjacent building. So one company we have. So what are the product? We are isolate the product, the determination of the product. After the determination of product, immediately the products go to the pharmacology studies. After that, it is going to the approval of the patents and the FDA. Then followed that, the products will be there to go to the market immediately. The companies are adjacently there. If any remarks or any other problems there, immediately come to that. Immediately come to the lab. We have analyzed immediately within 1 month. So if anything is happened, immediately we have give the solution. So this is a Korean atmosphere. So why I'm telling is many of the India universities are there, but the research lab is so very far away. So many of the research labs are not the collaboration with in the universities, and companies are not collaborate with the universities. Universities are not collaborate with industries. So many gaps are there. So my experience from the Korea and Japan, such type of things, they are having a mutual understanding. So for the production of the compound, for the serve to the society people. This is one thing I feel in that Korea. So next one, when I go next trip next year, after 2 years complete, I went to that Malaysia. So University Malaya, Kuala Lumpur. It's a very good interest, a very good university. They are providing a lot of fund in this university. I have received 1.5 crore fundings for the 6 projects. Of that, nearly 6 to 7 people work under my guidance. The PhD scholar, master scholars, they are working under with my guidance. In addition that, there is there is also some research gap is there immediately when the university is now complete work completed immediately. Every month, university asking some question to us, as well as they are providing free funding. So many sources are giving to us. So based on the utilization, we have published more paper. In this nearly 35 papers I have published from this university affiliation. So followed that, I have worked a few months in Thailand for the research. 6 months, around 6 months, I worked in the Thailand. Followed that, I worked 2 years in Ho Chi Minh City, Vietnam, Tan Du Stang University. So here also is very good research environmental is there. People are more interested. More people are more interested to do research. They work on the morning up to the evening. They Saturday, Sunday they are working. So nowadays I feel that we have that very strong in the reading the books, all the things. But the research we are very lacking. We are not properly concentrate on the research work. But examination point of view, our Indians are mark aspect. Our Indians are very strong. There's no doubt about it. But the research aspect, I'm really very disappointed regarding the research aspect. We are not concentrate properly. The students nowadays, the students are so much laziness in the research aspect. When the Korea and Malaysia, the people are always maximum times they will sleep in the lab, but we cannot see this type of things in Indian environment. Very few lab like IIT, such type of things. So all the students to work forward to dedicate to that research contribution in the different angle, then only we can get the successful research. This is my experience, Dr. Purushottam sir. Thank you.
Thank you very much, sir. Your experience should inspire the youngsters. That while I asked to share your experience, that's really beautiful experience. Thank you very much for your presence. Wonderful persons in BioYojana 2021. Now the section is going to the audience. Okay. Now you can ask your question to Dr. Bakrudeen sir. Kindly raise your hands.
Okay, sir. I think student, I give my things, email ID, phone numbers are
Okay.
there. If they
Okay.
have questions, please anytime you can contact me. I really help to that all the students. If the in terms of any other help in the research, I'm ready to help to all the research scholars, students, or research scholars, master degree students, undergraduate students. Anyone, anytime contact me. It is always welcome you, because my intention is work on the research. We need to get the more product, this type of things. Thank you one and all. Thank you, Dr. Purushottam sir, providing these opportunities. This I am feel very good. Very good. Last few days, the people are attending this conferences. What a very wonderful. This is objectives and goals are very interesting one. I thank to that Can Bio R&D Solutions CEOs, Dr. Purushottam sir. Thank you one and all.
Thank you, doctor. One more question for you, sir. Doctor
Yes, sir. Yes, sir.
Sweta, now you can shoot your question.
Sir,
Yes, Sweta.
Okay.
sir, for gymnic acid derivatives, can we use it for different people of different aspects, ages, size, and all?
Yeah, yeah,
Gymnic,
yeah, yeah.
Yeah, because there are some products that have been restricted for some people based on their health condition. But why is that so, sir?
Yes, yes, yes. Sweta is very good, Sweta. Sweta, actually, the gymnamic acid is not only nowadays, the first research paper,
Yeah.
when first research paper is, I have the proof, nearly 1874. So first paper is published. Nearly more than 100 years, more than 150 years are starting the research in this field. Nearly in the Gymnasium University plant working around nearly 17,000 papers are published in this one. So that much potential plant material, as well as here nowadays also the daily life in the village side, the people are daily using the plant material. One thing is immediately if you are taking large quantity of leaf material or anything else, it having the you feel hungry. You feel hungry. That's why you have to take 2 leaves or 3 leaves like that. Based on the local people instruction, you follow that one. Another one is the glycolic acid compound is FDA approved compound. They are clinical trials successfully done, and that the glycolic acid is now is available in the Sigma. It is some expensive compound, but if you are request to the filing purposes, definitely they will contribute to us. Like the no side effect, it is approved drug only, but that you need to take the quantity aspect. You have to ask to the doctor or the particular scientist. If a large quantity we are taken, so what happened? I feel that I am taking the one day when I was working the research, I was taken nearly 10 to 15 leaves. Immediately I have feel very hungry. If
Yes, sir.
immediately after that also I will not complete that hungry. So that much it absorb the carbohydrate immediately. That's why if the another important proof, Sanmuga Sundaram et al. This IABMS Taramani campus, these people and the group members are 1977 to 80. They have done very excellent research. All the parameters they checked, like enzyme studies, like any other side effect or any other what are the things happen in the intestine, what are the happen in the liver. So many peculiar studies are done in the IABS Indian Biomedical IABMS Taramani. The Sanmuga Sundaram et al. That the group member is Sanmuga Sundaram et al. They are leaf material. Then the another animal experiment, the completely screen without the plant having no side effect like that. So based on this one, I am telling that it is valuable, no side effect, but we need to aware, no need to take large quantities. With limited taking very small quantities we are taken means it is very healthy one. Okay, thank you, Sweta.
Thank you, sir.
Okay, thank you, Dr. Purushottam sir.
Thank you, sir. Thank you for this wonderful presentation, sir. Thank you for your presence in BioYojana 2021, organized by Tanvi organization. Thank you once more, sir. Now, Soundarya, you can give a vote of thanks to Dr. Bakrudeen, sir.
Thank you, sir. Thank you very much for sharing your profound knowledge with us. Your session was full of valuable information on secondary metabolite production. Thank you so much, sir.
Thank you, Sweta.